Well, welcome to our NextGen Ellis Fischel lecture this afternoon. I'm Jeff Bryan, and it's my great pleasure to introduce our speaker today. I don't think she needs much of an introduction to this group. Dr. Carolyn Anderson has joined us here at the University of Missouri after 30 years of very well-recognized radiopharmaceutical research. It was my great pleasure to meet her in late 2003 or early 2004 when I was a graduate student here at Missouri. And Dr. Mike Lewis, my mentor, and I traveled to Washington University in St. Louis to discuss my master's project with her, which tested a hypothesis that she had that copper-64 might be more toxic than just its radiation properties. And although our experiment did not support that hypothesis, I learned a ton from the process and have really enjoyed my career in radiopharmaceuticals since. And Dr. Anderson is a well-recognized luminary in the field. She's received numerous awards, including the Michael J. Welch Award from the Society of Nuclear Medicine in 2012. She worked with Dr. Welch at Washington University in St. Louis. She's a Distinguished Investigator Award from the Academy of Radiology Research in 2014, and the Paul C. Ebersole Award from the SNMMI for Outstanding Achievement in Basic Research in 2020. She is the Simon-Ellebracht Professor in Medicinal Chemistry here at the University of Missouri and a Professor of Radiology. And her unique expertise is in inorganic chemistry, radiochemistry, and medical applications in cancer and other diseases. And as you'll see in her lecture today, one of the molecules that she studies has application in cancer as well as hematologic disease. And I think as we develop a larger and larger catalog of these targeting agents, we're going to see they have multiple applications across many chronic disease fields. And so learning to collaborate and to share these is going to be very important as we go forward. And I will not take up any more of her time, but Carolyn, thank you for speaking with us this afternoon.
Great, it's great to be here. I'm going to talk today on really a single radio tracer labeled with a couple different radionuclides and show you sort of the versatility, as Jeff mentioned, for one tracer in very different diseases, which are cancer and sickle cell disease. So I'm going to talk first. I would really like to tell all of you about what we're doing with molecular imaging and theranostics here at MU. So I'm going to start with that, and then I'm going to talk about the applications of targeting very late antigen 4 as a theranostic for metastatic melanoma. And then I will end on a project that I'm still working on with people at the University of Pittsburgh on using the same molecule to image vaso-occlusive crisis in sickle cell disease. So the reason, and I have to say, I'm so thrilled to be here. It's coming up on a year already. I can't believe it. Time sure flies. And it's great that we're coming out of the pandemic so I can actually meet with some of you guys in person again, rather than, the Zoom. And I look forward to these seminars being in person or at least hybrid in the near future. So I think, the reason that I'm here is because the University of Missouri, or Mizzou, has so many unique resources, and we have pretty much everything we need to go from benchtop to bedside and looking at novel ways to treat disease. So what the fun part is for what I get to do, and I get to work with all of you to do, is to connect all the dots of all these wonderfully unique resources. And these include, and I'm going to try and get my laser pointer here. So we, as of course, working with MUR and the production of radionuclides. This is our center that I'll talk more about, working with the CVM, School of Medicine, and NextGen, and how all of this really, we have pretty much everything we need to do to translate new agents into clinical trials in humans and in dogs, for that matter, and cats. For the molecular imaging and theranostics that we're all developing. I just want to give a shout out to the University of Missouri Research Reactor and Dave Robertson, who really, I know, worked hard to get both my husband and I here. And, MUR it's a unique national resource. It's really, it's a main reason why I'm here. This reactor has been around for well over 50 years now, and it's going to be around for a lot longer. It operates 24 hours a day, almost 7 days a week. There are many staff. They work very hard. They worked all the way through the pandemic with no break at all. And they are the sole Western Hemisphere provider of 5 nuclear medicine isotopes. So I hope everybody appreciates the gem that we have here in Columbia and with the university. And so we're located right across the street from MER. So this works out very well for us. And we get the benefit of being under their radiation safety auspices, which is great. And when we need radionuclides from MER, they just walk them across the street. And I'm fortunate to work with Jeff Smith as a co-director here of this of our center. And right now, if you ever come and visit us, it's got a name, the International Institute of Nanomolecular Medicine, but ultimately we hope it's going to say Molecular Imaging and Theranostics Center. So that's the goal, maybe later this summer if all goes well. And so what are the resources that we're going to have? And we're just calling it MITSI for Molecular Imaging and Theranostics Center. So we have lots and lots of synthetic chemistry labs. So everything we do really, in addition to, we get the radionuclides that other people produce like MER and other places like WashU and University of Wisconsin, they come in and then we do synthetic chemistry. We have radiochemistry labs. We have a bioanalytical lab as well as a 400 megahertz NMR. So we have everything we need to do for both synthetic and radiochemistry. Jeff Smith runs the radiopharmacology course. So we are also providing services to people and inviting, training people, the students and the postdocs here in doing the biochemistry and the cell-based assays. We have all the equipment. We have, well, we're actually down a staff person right now to perform the studies, but we are still managing to get them done. We also do the biodistributions. Lisa Watkinson and Terry Carmack do those. And hopefully by December of this year, we are going to have our preclinical imaging suite open. We will be having a Bruker Albira SI, which is a trimodality scanner, PET spec CT. This is all for rodents, and a rodent biospec 3T MRI. And we'll have, again, staff that will be able to work with people to do studies. We'll have full quantitative analysis capabilities that we can either do for customers or we can train your students and postdocs to do their own data analysis. This is what our suite is going to look like. It's going to be in the lower level of the MITSI building. It's about 1,500 square feet. And it's going to be, we're going to keep it as a barrier facility. So when you come in here, you're going to have to gown up and wear all the PPE so that because we have the vivarium rooms here. And so if you do want to do studies, you can have your rodents either transferred in here or ordered in here. We'll have procedure space, and then we'll have the 2 scanners. And all of what we're trying to do is to leverage our experience that already has existed at the University of Missouri and MER now for probably 35, 40 years. They've been making radio tracers, and it's probably the only place in the country where they've developed 3 agents that have been FDA approved and are still being used in as part of the standard of care in patients with cancer and other diseases. I do want to point out the TheraSpheres, which this only had limited FDA approval through a humanitarian device, but due to COVID, they were preferring to do the TheraSpheres rather than going in to do surgeries and inoperable liver cancer, not even surgeries. I think there was some other procedure that was a bit more invasive. And so this got expanded approval in March of this year. But if you look, all of these were approved, 20 plus years ago. And so the goal here is, to kind of bring back those glory days, develop some new agents that, we can now license to big companies and big pharma as we have with these other agents. And then I also just want to give, for those of you who may not be familiar with what Theranostics is, and Theranostics is not just what I'm going to show you here with radionuclides. There are other examples, but in area of radiopharmaceuticals, theranostic is one agent. In this case, it's this dotatate compound. It's a peptide, cyclic peptide with a chelator on it, and we can label it with the radiotherapeutic radionuclide lutetium, or we can label it with gallium-68. So we can do both radionuclide or radiopharmaceutical therapy with one compound, or we can do imaging, diagnostic PET imaging with another. So both of these are now FDA approved, with the therapy agent being approved in 2018. I believe NetSpot was in 2016. But I just want to make sure everybody is aware that the lutetium is produced at MER, and it is the sole producer for this FDA approved product. And then I just also want to give a shout out to the College of Veterinary Medicine and the Pet Center that Jeff Bryan runs. And this is our bridge. So we do all the rodent work here at MITSI. We'll be doing all the rodent imaging, the theranostic development, testing them first in mice and rats. And then if we have something where there is a spontaneous disease in companion animals, we can bring the technology over to the CVM, and they will work with us there. And they are already quite busy over there with a lot of collaborators that are shown here, doing both imaging, and then I think they're also doing therapeutic agents as well. So I want to give just kind of a overview of what it is we do when we're developing molecular imaging and theranostics agents, and where is all this going to happen on the campus. So we start with radionuclide production, which is going to be at MER or other places for the PET isotopes. We do the chemistry and the radiochemistry. We work closely with the molecular interactions core, which is wonderful. It's here on campus. So we've done sort of a hybrid where they make parts of the molecule, we make parts of the molecule, and then we do all the radiochemistry here at MITSI. The radiopharmacology core is there to do all the in vitro studies. So once we make these radio tracers, we can evaluate them both in biochemical assays or in cell culture. And then we'll have the preclinical imaging suite here to do all the rodent imaging. We do rodent imaging and biodistributions, but we also want to validate what we're seeing in the images ex vivo. So for that, we will work with the cellular and the CIC, which now I can't remember, immunobiology, cell and immunobiology core, which is also wonderful. We get all our cells cultured there, and it's just seamless. And then if we have good imaging and validation in the mouse models, we can then go over to the CVM, or we can go into NextGen. And I'm really excited. We had a really great meeting this week with people who are working in some of these oncopigs. So I think there's going to be a lot of synergy with what we're doing and the pig colonies that they're going to have at NextGen. And then finally, the major goal is to do first in human imaging of something that we've developed that's novel, or, also working with companies who want to do clinical trials, and all of this can happen at NextGen. So one of the major efforts that we have ongoing right now is to recruit a clinician scientist who's either in the fields of nuclear medicine or radiation oncology that has some experience in theranostics, so that can work with us in addition to the people who are already in nuclear medicine to bring these agents into these clinical trials. And then oftentimes, we do have to go back to the drawing board, because what looks great in mice and maybe even in dogs doesn't necessarily translate to humans. So we might have to tweak the chemistry. But this is sort of everything we're doing. And this is what we're going to be doing here at MITSI, but we're going to be working with many of you who are listening to this to fulfill this whole cycle. So now I'm going to talk about the projects that we're working on, and this is just it's kind of a subset of what my lab is working on. But we've been working with this particular biomarker called very late antigen 4, which is an integrin a cell adhesion molecule, alpha 4 beta 1. And so the integrins they're non-covalent heterodimers, they're transmembrane, as you can see here, transmembrane proteins. They're expressed on various types of cancer cells, such as melanoma, lymphoma, and multiple myeloma, but they're also expressed in immune cells, and what I'll show you later on, reticulocytes as well, which is important for some of the hematological diseases. The natural ligand for this particular integrin is VCAM1 and fibronectin. And this particular integrin plays a very important role in tumor growth, angiogenesis, and metastasis. It promotes adhesion and migration of cancer cells, but it also, and I'll talk later about the sickle cell disease, it works in so many other diseases as well. And I actually gave a talk this morning for a NIAID symposium on imaging and infectious disease, and we've done a huge study to look at monitoring drug efficacy with the same compound in tuberculosis. And what else is very interesting about this particular integrin is that it's always present in many types of cells in a low affinity state, but when it's activated, when there's some sort of insult, when there's cancer, or there's inflammation or infection, then we get this high affinity state. And what we're imaging is specifically this high affinity state, so that cuts out a lot of the background. So LOP2A, which is the compound, this is the parent compound here. It was discovered by Kit Lamb at UC Davis, and he had a real tour de force paper. When I saw it, I immediately knew I wanted to work with this compound. So I've been working with it for that long. I think we started working with it in about 07 or 08. And it was discovered, what Kit Lamb is very famous for is the 1-bead-1 compound combinatorial screening that he does. So he created a library that has 5 × 10⁻⁻10 compounds, and he changed a bunch of amino acids. He put in a natural amino acid and peptido and D-amino acids, various things. And then he screened this library and blocked it with a compound that binds the VLA4. And he came up with a couple different compounds. So you go with 5 × 10⁻⁻10, you narrow it down to a workable number. And then out of this, he found LOP2A, which is very, very high affinity and highly specific just for this integrin. I don't know if anybody else has worked with integrin compounds, but they tend to bind a lot of other types of integrins, but this one is highly specific. And he first screened it with a dye conjugate of the LP2A and showed that it bound to VLA4 in this MOLT4 tumor. So we decided to start looking at it in melanoma models. And so we did make some modifications to his original compound. We put in a PEG-4 linker, which gave us some more water solubility. And what we found is you can put this R group here can be pretty much anything. And we put dyes on there and chelators, bunches of different chelators, and they all work very nicely. So I work with copper-64 a lot. I thought I had that on here. No, I don't. But copper-64 has a 12.7-hour half-life, and being in Columbia, Missouri is great. It's almost as good as being when I was at WashU in terms of getting the radionuclide where they produce it, but they drive it 2 hours here. So they produce it on a Tuesday morning, and we get it by around 11 o'clock in the morning here at Mitzi, so we can start working with it right away. Where when I was in Pittsburgh, we had to have it shipped overnight by FedEx, and we lost, with 12-hour half-life, you lose a lot of activity. So very happy for working with that again and getting it same day. We've been working in the melanoma. We initially start working with the B16F10 model. It's a very common model, and the tumors can be injected either sub-Q. If you inject them intravenously, you get a simulated metastasis where the tumor cells sort of go through the bloodstream and then seed in different places. So in terms of imaging this, and this was work we did several years ago now and published back in 2014. But basically, here's the B16F10 sub-Q model. We get, it's a very clean image. We just really see tumor bladder. That's just because the compound is excreted in the urine, and we really don't see much else. And we get very nice tumor to contrast ratios. If you do the intravenous model, and we also have transfected the B16F10 cells with firefly luciferase. So when you do bioluminescence imaging, you can see this is actually not, it's a bone metastasis, not a brain, but we get lung mets. We get, I think these are bone mets here. We also got metastases on the ovary. So all of the mets that we saw just from the tumors themselves, we were able to pick up in the image with the copper 64 LOP2A. And again, there's very little background with this compound. So it looks quite good. So, so we published all this, and I'll show you some therapy studies we've done. But you know, B16F10 it's an old model. It's not a very representative model of what happens in human cancer, and it lacks this BRAF V600E mutation that's found in greater than 50% of metastatic melanoma. It also has PTEN, which is again not necessarily representative. So working with Walt Storkus at University of Pittsburgh, he created some cell lines for us, and they were derived from the BP cell line that Jennifer Wargo's lab published. And this particular cell line it does have the BRAF V600E and it's PTEN negative. It responds to BRAF inhibitor treatment. So that's the parent line. Walt's lab conditioned the cells to be dabrafenib resistant. So they're resistant to BRAF inhibitors. And then his lab also transfected the cells with CD49D, which is the alpha subunit of the integrin alpha 4 beta 1. And then he produced the BPR alpha. So we have, I haven't been working with the parent line, but we've been working with this BPR and the BPR alpha. We've done, and so we're working on a manuscript right now. It's almost ready to be submitted, where we're sort of characterizing this pair of cell lines so that other people can work with it if they're so interested. We showed, and we did some comparison with the B16F10. So the BPR, we actually get better internalization, slightly better. I know it's not necessarily, I guess it is significant here. So we get better internalization compared to B16F10. We can block it, so we know that the BPR alpha, that alpha 4 is working, and we can block it with our compound. And we get essentially no internalization in the BPR line that does not have the alpha 4 subunit. And we also did a receptor binding assay, and we didn't get quite as high a binding affinity as we saw with the B16F10 line, but we did see more sites on this. So it is definitely slightly different than the B16F10 in how it binds to our compound. And here's some of the PET imaging and biodistribution studies we've done so far. So this is a biodistribution at 24 hours post-injection. We're seeing significantly greater uptake in the BPR alpha versus the BPR. So we know that, again, we're getting higher uptake. We do see a pretty good amount of uptake in the BPR itself. And we think this could be due to the immune cell component, because I know melanoma, at least B16F10, has a pretty strong component of immune cells in the microenvironment. And we think that this may be contributing to that uptake, but nonetheless, the BPR alpha is higher. And here's the images at 4 hours and 24 hours. And the BPR alpha, although you can see the tumors in the BPR line, they essentially are background by 24 hours, whereas you do see more retention of the copper LOP2A in the BPR alpha versus from 4 hours to 24 hours. Although there really isn't a lot of, this is the ratios, but they're in the SUVs, we definitely see at both 4 hours and 24 hours, we see higher uptake, that's statistically higher, and the BPR alpha than the BPR. So this is just to summarize, we've now, in terms of melanoma imaging, we've shown that the copper LOP2A works very well. It has high affinity for VLA4. The B16 model we've been using for proof of principle. And we now have this new model that we've gotten the cells here at MU, they're growing here. And we will be working with this model to further characterize it, especially for therapy. So going into therapy, so we can label the lutetium or the LOP2A with lutetium-177, which is and the entire time I've been working with this isotope, even at the University of Pittsburgh, we always got it. We got it straight from Murr, and then we had to kind of go with the middleman with the Department of Energy, and now we're back to getting it directly from Murr, who just brings it across the street to us. So lutetium-177, if I haven't mentioned it, has a half-life of about 7 days. And so we've done, when we were at the University of Pittsburgh, we looked at the biodistribution and we saw very high tumor uptake. But for some reason, this particular version with the DOTA LOP2 or the DOTA lutetium chelate definitely has higher uptake in the spleen and the bone marrow than what we saw with the copper compound. And it's not surprising in some ways because we know that the very late antigen 4 is going to be in these immune cells that are in the spleen and the bone marrow. We also showed that we can get a modest therapeutic effect with the lutetium LOP2A and a modest increase in survival with this. So what this kind of told us is that although this compound is, it's reasonable, it's not the best compound we could have. And so we're now looking at like, how can we better match? As you can see, the tumor uptake just clears out very quickly here, even by 24, 48 hours, there's essentially very little tumor uptake. This is a small molecule. The lutetium has a 7-day half-life. So what we want to do then is extend this without having to go to large antibodies or nanoparticles. And a way to do that is to add these small moieties called albumin binders. And basically, these were developed, some of them have been developed by Dario Neri in Switzerland, and this particular one is one of his albumin binders that other groups have used, such as Roger Shibley and Christina Mueller have shown this worked well with folate. So in another project, and this is the only slide on this project, we showed that when we took a different small molecule that targets PSMA and we added this albumin binder, we got amazing results. We really literally went from very low tumor uptake and fast blood clearance, and we multiplied the tumor uptake by about a factor of almost 20. And we slowed the blood clearance, we got very high tumor uptake. And then when we looked at the therapy studies, you can see that the non-albumin binding molecule essentially had no efficacy versus control, but this albumin binding molecule showed almost, we had 80% survival out to that was about 120 days, but we actually went out as far as 200 days, and we were seeing about 80% survival. So we wanted to give this a try. So this was the first, one of the first things we started working on when we moved to MU. And Michael Punn, who's been a real trooper, he started with me at the University of Pittsburgh. He was probably the only person in my lab who knew back in January of, or even before that, of January of 2020, that my lab, that I was considering moving here, and he came and worked with me anyway. And so Michael got the lab set up here, started working with Fabio Galazzi in the Molecular Interactions Core, and together they came up with a strategy to make these two compounds. And so the LOP2A with DOTA is slightly different than the one that I showed you that we previously published. I think it's going to be it's actually a little better because it has better coordination with the lutetium, having 4 carboxylates. And so we made this as the non-albumin binding, and then we have this para-iodo benzoic acid molecule, which is going to be the albumin binding molecule. So this is the initial radiolabeling studies that Michael did, showing that both compounds bind and we can get high specific activity and high molar activity with the lutetium from Myrrh. And then Von Ho, who's a postdoc who got her PhD here at MU, she is doing all the receptor binding studies. And this is the non-albumin binding, has very high binding affinity. But the PIBA, the albumin binding molecule, has somewhat lower binding affinity, but again, it binds more sites than does this one. And this is always kind of confounds me, and I'm never quite sure what's going on with that. But nonetheless, they both have high affinity for VLA4. So we then moved into biodistribution studies. We just started with the B16F10 model because we have lots to compare it with, and we did the 2 radio tracers, as I mentioned, the albumin binding here and the non-albumin binding, and then we did blocking studies, and we just used the LOP2A with PEG4 on it as a blocking agent. We did several time points first out to 96 hours, and then we actually went back and did longer time points with the albumin binding, and we took several organs as well. So ultimately, we want to be able to do the imaging, not quite ready to do that here yet. So we just initially started with biodistribution. So I'm just showing you kind of a snapshot of what's going on, just so you can see what the difference is between the non-albumin binding and the albumin binding. So we get high tumor uptake, but it clears out quite quickly. And this again is out to 96 hours. So here's the tumor uptake. The blood and the muscle are essentially very low by 96 hours. With the albumin binding, honestly, it wasn't quite as good as I was hoping. We saw it kind of the uptake increased up to 24 hours, but we still got this high blood uptake. And then we didn't see tumor to blood above 1 until 48 hours. And we're only this is out to a week. So, but the good news about this compound is that the tumor uptake does retain at 1 week, where here it's essentially almost baseline by 4 days. And then these are the tumor to non-tumor ratios. This is sort of how we look at things in terms of a therapeutic index. Now here's the bottom is the non-albumin binding, and pretty much similar tumor to kidney and tumor to muscle. The tumor to blood is very, very high for the non-albumin binding compared to the albumin binding. So again, this is sort of where I'd like to start working with this molecule to tweak this and see if we can't improve this, getting it out of the blood a little bit faster, and it could mean going with a different albumin binder. And so this is what we've done so far. Michael Punn is an amazing medicinal chemist. He's working on more compounds. He's working with a modular click chemistry compound. So he's actually synthesized this portion here with this, there's going to be an azide on one side and a DBCO on the other. And where you can we can then work with other people if they want to use this click chemistry to look at their own molecules. We can give them pieces that have this albumin binder and a chelator on it. We're also looking at other dyes. And so Sean Chen, who just recently moved to Singapore from NIBIB, he has published on the Evans Blue. So we're going to take a look at that, both with the click chemistry and the non-click chemistry. And so that's kind of what he's working with so far. So to summarize, on the LOP2A with lutetium, albumin binding definitely works. We're getting slower blood clearance, somewhat increased tumor uptake, but most importantly, retention in the albumin binding version. We are going to be doing some SPECT-CT imaging as well as therapy of these albumin binding agents in both the B16F10 and the new BPR model. We're going to work to do the in vitro and in vivo studies on his next generation molecules. And we're also really excited to start working with terbium-161, and Heather's student Patrick Bacolo just did his first, we did our first labeling studies with Patrick last week. And so maybe at a future seminar in some venue, we'll be telling you how the terbium-161 works as a therapeutic isotope. And it should be better than lutetium. It has all the features of lutetium, but it's also going to have these low energy electrons that will hopefully provide additional therapeutic benefit. Okay, so in the last about 15 minutes or so, I'll talk about a project on sickle cell disease that I started working on probably about 6 years ago at the University of Pittsburgh. And just a little bit of background. So when I was 9 years at the University of Pittsburgh, but I was 4 years in radiology, like 5 years in radiology and 4 years in the Department of Medicine. And in 2016, due to changes in department chairs and various things, I was sort of invited to move over to medicine. And the person who was recently named chair, Mark Gladwin, his main area of research is sickle cell disease. And Mark built a very big critical mass of people that work on this disease, which I knew nothing about really in terms of, I think we all know what it is, but I knew as much as everybody else here about what sickle cell disease is. Just to give you an overview, it's a genetic disorder. It affects about 100,000 Americans and a huge population in sub-Saharan Africa. It's characterized by increased cellular adhesion from chronic inflammation. And this causes these very acute painful events called vaso-occlusive crises, and they also called vaso-occlusive episodes. And about 80% of patients who have sickle cell experience these VOCs, anywhere from 1 to many times a year. But there's really no way to image or diagnose VOC or its severity. So it's sort of this nebulous thing, and nobody really knows exactly what it is or whether patients even have it, because there's no way to characterize it or detect it. And so what Mark, when he, I think it was back in 2015, he asked to meet with me, and he said, we need a way to image this vaso-occlusive crisis. There's a small group of patients in Western Pennsylvania, about 250 that I think are regularly seen at the clinic. And I think he had just lost a 16-year-old girl. And again, there's usually no warning when people die. The VOC turns into something even worse, like acute chest syndrome, which is fluid in the lungs. And people just die, and they have no idea like how bad their disease is. And so I worked then with Enrico Novelli, and he is just one of the most dedicated people I've worked with in terms of how he deals with the patients that he has. And he's so, very focused on finding some way to help his patients. And I think imaging is this is one of those cases where imaging really will help. I mean, cancer imaging also helps, but this is where there's just no options. There's no biomarkers, there's nothing. Interestingly, I ran across this article, and it was like a little over a week ago in the New York Times. And it was an article about a woman who she's completely debilitated with pain from sickle cell disease. And this was an excerpt from the article. Biopsies can detect cancer, x-rays of broken bone. There is no definitive clinical test to determine when a sickle cell patient is suffering a pain crisis. Basically, this is a doctor from Johns Hopkins who said there's no objective measure. The gold standard is the patient walks in and says, I'm having a crisis. It'd be like a cancer patient walk in and say, I'm having a tumor, or I have a tumor. That we would all think that's insane. So, so this is a disease that has been, just not studied enough, and there's just not enough out there to help people. So what we have been working on is, or what we know about the disease at least, is that sickle cell vaso-occlusion involves hyperadhesion of red blood cells and leukocytes to inflamed vessel endothelium. And so this is what we set out to image. Are these vaso-occlusions caused by the sickling of the red blood cells? And a postdoc from my lab, Leah Niran-Schutte, who's now at UCLA, Lydia Perkins, who was a joint postdoc between my lab and the Novelli lab, and then Linda Eyrig, who works for Enrico. And so Lydia is now at Carnegie Mellon. And so interestingly, when we started working on this, we knew integrins they could be good biomarkers. And the initial integrin we worked with was alpha V beta 3, which I'll show you something we did with that right at the end. But I kept reading about it, and I was like, VLA4 looks to be a good biomarker for this disease. And it's found on leukocyte adhesion, which are the white blood cells, but it's also found on the reticulocytes. And we initially thought, well, we're just going to see blood uptake and we won't actually see anything. But we went ahead with it anyway, and people were very skeptical as to whether or not this would work. So fortunately, and here's our favorite compound, copper LP2A. Fortunately, there's a mouse model of sickle cell disease, which is Townes mice, and they were developed by Tim Townes, who I believe is at the University of Alabama, Birmingham. And so they're transgenic mice. There's sickle mice, the SS, they're called also M21s. M22s are the non-sickle littermates, also called AA. So we were able to, we had some of the people who do sickle cell research at Pitt have breeding colonies, but we had our breeding colony at Jackson Lab, and we would just order in the mice when we needed them rather than breeding them ourselves. And so what we did was longitudinal imaging, where we did baseline imaging with the VLA4 tracer and imaged at 4 and 24 hours. And then we gave a minute, this is actually 0.5 micrograms per kilogram, so minute dose of LPS, which is causes inflammation. We injected it IV, and then so we injected the LPS and the tracer together, and then we did the imaging study. And again, I was really like not sure, especially this co-injection of LPS with the tracer. I was kind of skeptical about that. We had Invion, which is a Siemens instrument for PET CT. So here are the images. Here's the non-sickle mice in baseline. They do have big spleens, but there's absolutely no change between baseline and post LPS. And typically we're looking at in the limbs, in the extremities, because that's where we're going to see the higher levels of uptake. So here's the sickle mice. The first thing we notice, these have enormous spleens, but you can see here at baseline, we still see some pretty decent uptake, but then post LPS, you can see the uptake is much, much higher. And we looked at both the femurs and then the humerus, which is the joint area. So I'll show the quantification of these data in a subsequent slide. But what we also did was we wanted to know, okay, so where is the LP2A binding? And so for that, we used a dye version of LP2A. We did flow cytometry, and this was all post LPS, and we compared non-sickle and sickle. We looked at reticulocytes, the immature red blood cells in the spleen, bone marrow, and blood. We looked at just CD45, which is a general marker for immune cells. And then we narrowed it down to the myeloid cells, B cells, and T cells. We definitely saw in the bone marrow, there was this increase, a significant increase in the sickle mice in the bone marrow. We saw a decrease in the blood, which we thought, I'm still not sure I can totally explain. We saw a increase statistically significant again of the myeloid cells in the bone marrow and B cells in the bone marrow and spleen. And then the T cells, we actually saw them drop in the sickle mice in the bone marrow, but otherwise we didn't see any differences in the spleen or the blood. So then the good news for sickle cell patients is there are a lot of new drug treatments. There also is gene therapy, and that was, if you read the New York Times article, like some patients are just not eligible for things like gene therapy. There's also stem cell transplants. So, but several new drugs have come out. And there's crizanlizumab, which was FDA approved in 2019, which is an antibody against P-selectin. Rivapanzol, I believe it's a small molecule drug. It's in phase 3. Voxelotor, which targets hemolysis, that was FDA approved. It's small molecule, approved in 2019. And L-glutamine was approved in 2018. So there are at least options now. And then hydroxyurea is a really old drug that was essentially the only drug for many years up until the last few years. So, but the problem too, I mean, these drugs, it's a great, wonderful thing now that we have all these drugs, but the problem is we don't have any way to know whether patients are responding to treatment. And the current paradigm is trial and error. The new drugs, especially crizanlizumab, we're looking at 100,000 per year. There's side effects, and that's actually being explored a lot and our mouse state actually helped explain some things. I'm not talking about that today. We're seeing in patients, and less than 50% of patients actually respond to any of these drugs. There's no precision medicine, so there's no way to image this. There's no biomarkers, not even serum biomarkers. And so it's just this, well, which drug should we give the patient? It's trial and error. They're waiting around a year to even know whether or not they're responding to the different drugs. So we chose in the mouse study, we chose to look at, we used crizanlizumab as our first drug, and we used a mouse version of that binds mouse P-selectin. But crizanlizumab actually, so P-selectin, what it does, it blocks the binding, or let's see what, I have it on the next slide. It actually blocks binding of leukocytes to the endothelial cells and platelets. And so that's how it works. And it was shown in a clinical trial that was published in the New England Journal of Medicine that it reduced the annual rate of sickle cell pain crisis by about 45%. So we were wondering, could copper LLP2A be an earlier indicator of response to this anti-P-selectin antibody? And sorry, it's interesting because I think VLA4 is downstream from the P-selectin. So they're kind of in this whole adhesion cascade. And so that's why we decided to try this one first. So here's the imaging study. We actually finished this up right when we got kicked out of the lab last year. So it was kind of amazing. We did a Hail Mary and it worked. And so basically, here's the study. It was very similar to the one where we just wanted to show proof of principle. So we did the baseline imaging. We only worked with the sickle mice. We did not work with the non-sickle mice. And then we gave either the anti-P-selectin antibody or we gave IgG as a sort of an antibody control that's not going to bind P-selectin. So here's the sickle cell mice at baseline, and then plus the IgG. So we see that there is, again, after and post LPS, forgot to mention that we were giving the LPS, we gave it all together. So we gave LPS, antibody, and tracer in one injection, or one after the other. I'm not sure if they were all in one injection, but they were all given around the same time. And then with the P-selectin antibody, we did not see this huge increase in uptake after the LPS. So here's all the quantification. There's a lot of data here, but basically this side of it is the first study we did showing that post LPS, we see an increase in the sickle mice where we don't see them in the non-sickle. And then when we gave IgG, the data were a little bit messier, but we definitely in the femurs, not in the humeri, but in the femurs, we saw an increase in uptake with just IgG. We saw not a statistical increase with P-selectin. And then here's just another way to show the data with the increase. And then with the P-selectin, you can see it brings it essentially back down to baseline. And this was published in September, I believe it was, in Blood Advances. So this was also another study we did. I mentioned that when Mark Lavin came to me, he wanted to do an imaging study. We went to Shenzhen and we got this PRGD2 because they had done several clinical trials that are cancer related. They'd also looked at cardiovascular disease and some other things. And it was already sort of, kind of ready to go to put into patients. So we just did one, it was a case study. There was a woman, she's 29 years old at the time. She had sickle cell disease, and we imaged her with PET MRI, both at baseline and then when she was in crisis. And she had her specific pain was in the thighs. So we really limited the PET MRI study to between the knee and the hip. And that was kind of the downside of the PET MRI, doing that rather than PET CT, but that's sort of what we did. And then what we saw, so what we did then was we looked at the MRI to pick the regions of interest. We looked at the blood vessels and the muscle as a kind of a reference point. And then we also looked in the bone. And we did the SUV measurements. And then we did SUVR. So here is the bone to muscle. This is, again, just one subject, so there's no statistics here. But we saw that there was somewhat higher SUVR, which is the ratio to muscle for the bone, and the bone to vessel. But we saw some higher uptake when she was in VOC than when she was in baseline. And here are the PET MRI images from that particular woman. She was a wonderful person. I was kind of fortunate enough to be there when she got her second scan, and Enrico was with her the whole time. And she gave us a lot of feedback. So when we do the next clinical trial, which should be hopefully starting soon, we learned a lot. And she was just a really brave person to do this with really no benefit to her at all. So to summarize, I think there's a major need to objectively measure pain crisis in patients with sickle cell disease, both to determine severity of the disease as well as monitor drug treatment. VLA4 is a viable biomarker for monitoring VOC that results from an inflammatory stimulus. And we have great data in the Towns sickle mice model, showing increased uptake of the copper LP2A in mice, femurs, and humeri that received LPS compared to their non-sickle litter mates. We also saw that we could image response to an anti-P-selectin mouse antibody in the sickle mice that received LPS. And we have this case report that kind of just shows us proof of principle that, yes, we're able to do a clinical study. And this is again at University of Pittsburgh. So what are the future directions? You might wonder, like, what would we do here, if we wanted to continue this? And at the University of Pittsburgh, they do hold now an IND, an investigational new drug application for LLP2A to do, and the human studies approval is in place. They were, again, slowed down by COVID. They're also getting a clinical coordinator on. They're hiring that person so that they can get this trial started. The second thing we're trying to do is there's multiple different triggers that cause VOC. We looked at inflammation. We also want to look at things like hypoxia and other triggers. Those are studies we can do here. We can order the Towns mice here. We could do some of these imaging studies. So Enrico and I have an NIHR1 that's being reviewed on June 10th, which by the way is today. So don't know, I'm sure it's all a done deal by now. But hopefully this will be reviewed positively, and we can continue what we've started. And the other things that we'd like to do here are developing new agents. And I think based on P-selectin, we looked at some peptides, didn't work very well, but we might go back to the crizolizumab and actually make this an imaging agent and see how that works. Another thing that I think is really exciting, and is that we could actually do a radionuclide therapy in these patients. Some patients are actually eligible for stem cell transplant. There have been cures. These are mostly the young people who are already strong. When they undergo stem cell transplant, they essentially get chemotherapy that wipes out their bone marrow, and it is extremely harsh. And I've read papers on this where it can actually kill getting prepared for a stem cell transplant can kill a patient. And so we think we might be able to do this much more safely with a therapeutic version of LOP2A labeled with either lutetium or possibly holmium, which was recommended by some of the people here at MU because it is a higher energy beta and has been investigated actually in some of these types of things like wiping out bone marrow. So this is something that I think I'm talking to people at University of Pittsburgh to see how we can get this started, because we think this is really exciting. So finally, I just want to end by, these are my collaborators and the people in my lab when I was at Pitt, and I'm still working closely with Enrico and Walt Storkus, who with the BP and BPR cell lines, and he's been a great immunology source for us. Ravi Patel is a radiation oncologist / PhD who I wish we could get here. He is really wonderful to work with, and he's the exact kind of person we're looking for as far as our clinician scientists. And Mike Bellavia is a student that I'm still co-mentoring with Ravi, who's opted to stay in Pittsburgh. Here's our group here at MU, which is growing, and this is only some of the people. I have to give a big shout out to both Vaughn, Michael, and Fabio, who's been really wonderful to work with, and Lisa and Terry, who are like superb at what they do in terms of the in vivo studies. And then of course the funding that we've received, and hopefully we'll get even more to continue some of this work. So with that, I'm happy to stop here and take any questions. And thank you so much for being here and your time.
Carolyn, that was a fantastic presentation. A really great, congratulations. I'm going to keep my fingers crossed for your auto-one. So I have a couple of questions. VLA4, as you indicated, is expressed on immune cells, particularly activated cells. Have you done any studies, especially when using this theranostic approach? Have you ever checked the impact on the immune response? I'm just curious. Does it have any deleterious impact on the immune response?
Well, according to the initial study that as far as I know, the LOP2A itself, so the cold compound, does not have a pharmacologic effect. And in fact, John DiPerzio from WashU actually, I think, looked at it as an alternative to this. I think it's 2100 or that compound that Kit Lam used as a blocking agent. So I don't think the cold compound has any effect. Now, definitely when you start radio labeling it and you're killing off you might be killing off T cells and that. So we've done some testing with that. And yeah, you're going to see toxicity at a higher level of activity. And in the case of stem cell transplant, that's sort of the goal. So it depends on what we're doing.
Yeah, no, that's great. And the other question that I have, as you know, stem cells are expressing VLA4, and clearly, when using radionucleotide for treatment, that's something. Whether you have any idea there, what the impact is, and the last comment that you make, ESMA has been working using bone marrow to generate chimerism as a way of modulating the immune response to treat autoimmune diseases as well as preventing graft rejection. And one of the issues with generating mixed chimerism, it is a challenge. Usually, clinically, patients are being subjected to close to 400 centigrade total body irradiation. And there is the notion is that you have to open the space. for stem cells that you're putting in. And given that stem cells expressing VLA4, I think this is something, your concept is going to be really very conducive to what ESMA is trying to achieve this. Instead of using total body irradiation, perhaps we could work together and try to see, could we induce stable mixed chimerism using this approach, which is going to be much safer and perhaps actually effective.
Well, I would love to work with you guys. I think sounds like a really interesting idea. And yeah, I mean, that's kind of the goal too. I mentioned chemotherapy in terms of ablating the bone marrow, but when I talked to Enrico about it, he said, yeah, they also do whole body irradiation. And this would be more of a focused, and you're also getting the spleen with this as well.
That was a great talk. Thank you so much, Dr. Anderson. We are out of time. Please feel free to email your questions. I'm going to stop the recording, and thank you so very much.